having me here. I'm very glad to be here. I got to drive a Da Vinci machine this morning, which is very exciting. And now I know where I want my surgery if ever I need surgery. I'd like to spend my time today talking about not my own research. Actually, it's sort of interesting going to DARPA. When I got there, everyone said, you know, you have to forget that you're a researcher. You have to stop doing research. And I thought, yeah, right. When hell freezes over, I'm going to stop doing research. But actually, I haven't done much research, and I haven't minded. It's been a very interesting sabbatical. So today, I'm going to be telling you about other people's research on a program that I started pretty much as soon as I got to DARPA, called the Big Mechanism Program. We just had an announcement come out for my next program, which is called Communicating with Computers. And we can talk about that a little later during questions if you like. Please ask me any questions at any time, interrupt. This is a seminar, it's not a speech. And it's really nice to be back in an academic environment where people actually do interrupt and ask questions, so please be sure to do that. All right, I'm going to talk about what big mechanisms are and how we get them. But first, I'm going to say a little about why we did it. Actually, no, I have to put this slide up. I love being at DARPA, mostly. There are sort of aspects of the job that are a little aversive, and this is one of them. Anyway, the reason for the Big Mechanism program is that we don't understand complicated systems. And it just took way too long for that insight to really make it through my thick skull. But we don't really understand complicated systems, and moreover, the way we do research and the way academic promotion and tenure committees work, and I could go on, actually works against us understanding big complicated systems. And so this is going to be a talk about technology to help us understand big complicated systems. But right at the end, I'll say a little bit more about roles that universities play. I can give you all kinds of examples of places where our failure to understand complicated systems bites. There was a flash crash on October 15th. US T-bills, government-backed securities, lost 7 standard deviations in their value in a period of 2 minutes. That's bad, right? Because the government actually has to foot that bill. We talk about dumping iron filings into the sea to promote algal blooms. Well, that probably would work at a local level. That is to say, we have good causal knowledge that iron filings will cause algal blooms, but what it'll do to the entire climate, we don't really know. We develop, as scientists, with methods that have really been honed for this purpose, we develop very local causal knowledge, high quality causal knowledge about local influences, small numbers of factors. How those things play out in entire systems are really hard to know. And in fact, I think for many of the systems that we depend on for our survival, we don't know what the effects will be of interventions. So we're at a sort of an interesting stage of human history, if you think about it, where we have causal knowledge that allows us to intervene, but not global knowledge that will allow us to say what the effects will be. And that's actually getting pretty dangerous. And that's one of the reasons that DARPA was willing to support this program. Now, all of this state of affairs, is exacerbated by what I call pull scholarship. It's a very medieval form of scholarship. I go in my office and I pull results into my head, and I try to synthesize those results in my head, and then I sort of issue missives. I write papers, small number of papers, about very local, about the one thing that I'm interested in. And why am I only interested in one thing? Well, because since Sputnik, at least in the United States, people are rewarded for knowing about one thing. They're not rewarded for knowing about everything. or trying to understand things in a systemic way. So I sit there getting more and more accurate knowledge about the one thing I'm interested in. And of course the literature is growing like crazy, but if anything, my ability to read it is declining because I'm getting old and I process information more slowly. So I was talking to someone from NIH the other day. Medical researchers these days read about 30 papers a month. Actually, to me, that seems pretty good. But the publication rates in their fields are 10 times that. Right? So, you know, the literature is growing like mad. And so what can I possibly do? I have to narrow and narrow and narrow, right, in order just to keep up in what my field is, because the rates of publications are growing. And then I publish another paper, you know, adding to the cacophony. And who reads the paper? Well, you know the answer to that. Nobody. Right? The government paid for it, you know, paid my salary, my students, the lab, and nobody reads the paper. Most papers simply aren't read by very many people at all. That's the pull model of scholarship. Right? And it's grossly inefficient, and it precludes systemic understanding of anything. And so we'd like to have something that I'm calling the push model of scholarship. And in the push model, every time I have something to say, it goes out into cyberspace and it is made sense of by machines. And those machines build models, not of the one little thing I'm interested in, but of how all of those little things fit together to give me a global understanding of some big system. And every single result is merged into those big models. There is no such thing as useless research anymore, or papers that never get read. Everything gets read by the machines, and its implications are immediately calculated. That's the push model. I push stuff out, the machines ingest it. Now, all we really need to pull that off is machines that could actually do the job, right? Machines that could build big, complicated models of very complicated mechanisms. And that's really what the Big Mechanism Program was set up to do. We're focusing on cancer biology, and a good chunk of this talk will be about cancer. Yes, please.
You stated the problem as we don't understand big systems, and what you said is that this push model allows computers to build models of big systems. Does it allow them to understand big systems?
Yeah. Let me put that question off, because there's been some very interesting discussion in the last few months about what kind of models we have and what kinds of reasoning you can do with them. And I think that'll probably get a little closer to what you, but give me a minute to get there. Okay, so here I am at DARPA. Everybody says, yeah, that's a good idea. Now, what system are you going to use? Well, DARPA is interested in national security, basically. And so they would prefer us to work in areas that have to do with socioeconomic factors, insurgencies, social systems of one kind or another, how cities run. We can't do that. And I think one of the reasons we can't do that is the language that humans use to describe other humans is quite hard to understand. But we do have an axiomatic understanding of molecular biology. When it all boils down, when you're done, you get down to the level of the molecules doing things to other molecules. And so naively, and a lot of this talk will be exposing my naivete, naively I thought we should try to understand complicated biochemical systems. And cancer seemed like a really good candidate for a lot of reasons. It is extremely complicated. I think there are about 400 proteins on this slide, and all of them have been implicated in colorectal cancer. It has an absurdly big literature. If you ask for, say, the ERK pathway, you'll get 184,000 results. If you ask for MAP kinase pathway in breast cancer, it's similar sorts of numbers. There's a lot out there. And the knowledge is completely fragmented. Because as I said, every paper talks about a tiny little part of one of these complicated systems. And also what we're discovering is that the knowledge is inconsistent and contradictory. So this is a study came out of Peter Sorger's lab at Harvard. We observed remarkably poor agreement, consistently less than 10%, among different databases regarding pathway components. Now, don't get too alarmed, but basically what it means is people call things by different names, right? They include some parts of molecular pathways and leave out other parts. And so if you look at two databases, you'll get a very different perspective on what a given pathway looks like. And so it goes on, what's a canonical pathways database specific? And then finally, this is a significant problem for mechanistic modeling because, and this is a problem we run into in big mechanism, you'd actually like to be able to go out to the biomedical resources and say, tell me what you know about this pathway, and be able to compare it to what some other database knows about the pathway. You'd like a machine to be able to do that, can't do that. Can't do that. Inconsistent terminology, inconsistent in lots of ways. So we decided to focus on cancer, and then entirely by chance, I was introduced to a man named Frank McCormick, who's one of the world's experts in a particularly nasty pathway called Ras. Ras is a molecule that is implicated in some phenomenal fraction of cancers, and I can't remember if it's 30% or 60%, but anyway, either way, Ras cancers are, they are bad. Ras for a very long time was thought to be undruggable. Ras cancers are the ones you don't want to get. So we decided, okay, Frank said, we will put the resources of the Frederick National Lab at your disposal if your program comes up with any interesting hypotheses about Ras. And we said, done, we'll do Ras. Because I'm a computer scientist, I don't know, one cancer is the same as another to me. Let me just, by way of sort of telling you about the kinds of objects that we're studying, this is a very common kind of picture of a molecular pathway. Every term you see, MET, RAS, is a protein. And these pathways are protein-protein interactions. And this is somebody's picture of the RAS pathways. And one of the problems is that if you go to Google Images or really any resource and you ask for RAS pathway, you'll get a whole raft of very different pictures. People are very selective in what they put in these pictures, and in fact, RAS, if you were to look at all the things that RAS influences, you wouldn't be able to put it on one page by any stretch of the imagination. But anyway, what happens is RAS is a protein. You can view it as a switch. The switch is turned on or off by phosphorylation or ubiquitination. And when it's on, it initiates these other signaling cascades. And if you look, they end up with cell cycle progression, apoptosis, which is the cell killing itself, transcription. Anything that ends up here could cause cancer, right? Because cancer is uncontrolled proliferation of cells that won't die, right? So you worry a lot about pathways that end up in those essential functions. And RAS is one of the big ones. So the problem is that when for some genetic mutations, the version of RAS that is coded for is stuck in the on position, right? And it's just signaling, and that's what causes cells to become cancerous. And so the search has been on for a long time for ways to either interrupt these pathways or kill the cells that have RAS turned on or turn RAS off. And until very recently, there was essentially no progress. I saw just the other day that UC San Francisco, which does a lot of work on RAS, in fact, that's where Frank McCormick is from, has finally found a drug that will bind to a particular notch in the RAS protein, and in at least some kinds of lung cancer in vitro will kill those cells. So that's good news. Anyway, that was sort of just a very brief introduction from a computer scientist who doesn't really know this stuff about the kinds of things we're studying. Now we're about 7 months into the program. And pretty much everyone in the program is trying to build this kind of thing. We start with journal articles, and from those articles we extract a bunch of stuff. Some of it is text, some of it is tables. We're not yet working on figures. And the text then goes into a reading component, and out of that reading component comes fragments of knowledge about Ras-driven cancers. Those then get integrated by a process we call assembly, which revises prior models about Ras-driven cancers. There's a model manager that takes care of the proliferation of models. And finally, at some point, we do some reasoning with those models. And the reasoning would be things like try to reduce the size of the models, try to figure out the parts that really matter, figure out where an intervention might work, figure out how models integrate with one another, things like that. So everyone's trying to build this kind of thing. And we thought this was really all there was to it. And that's why the subscript here is my journey from naivete to realism. We've learned a lot in the last 7 months. So getting to your question about understanding these things, I'd like to spend a few minutes now talking about what's a model and what kinds of model machine reading might actually build, and what kinds of understanding you would like to have of these complicated mechanisms. When I started this program, I thought this was a model. This is the kind of thing you see in all of the biology papers. And it's really just another version of the picture I showed you a moment ago. Up there you see Ras in its unphosphorylated state, Ras-GVP, and then it goes through this process where the switch turns on and all these signaling processes are initiated. I thought that's what we were trying to build in big mechanism. And actually, building a really big, complicated one of these, as opposed to a sort of a sketch like this one, would be a valuable thing to do. We know that the situation is a lot more complicated than is here. We know that the knowledge is out there. It's just nobody's built the whole picture. So that would be a useful thing to do. But there's just an enormous amount that this picture does not say. If this were the family tree of monarchs in Europe, you might be able to reconstruct some of the history of Europe from it. But you see, you'd have a lot to fill in, right? And that's not a bad analogy. If this were major interchanges in a freeway system, you could sort of see where problems might arise, and you could sort of see how closing one thing might produce backups or, alleviate problems. But there's a lot that this picture wouldn't tell you. One of the things the picture doesn't tell you is anything at all about dynamics, or perversely, what the medical people call kinematics. Right, which is the time course of all of these chemical reactions. Right. So they tell you, for example, that there's this cascade, RAF, MEC, ERK, but they don't tell you anything about how much RAS, how much RAF, how much survives as you go on down, how long it takes, what the reuptake rates are. It doesn't tell you any of that stuff. So while this kind of sketch isn't a bad thing to try and extract from the literature, it is, after all, sort of a roadmap. It isn't everything you would want. So then you jump down to a very much lower level of understanding of complicated mechanisms. This comes out of Walter Fontana's lab at Harvard. And what Walter does is he says, well, we will express the underlying molecular biology by talking about the objects and where they bind to one another. So axin is an object, and it has a binding site. And beta-catenin is an object, and it has these binding sites. And I'm then going to write a rule that says if this thing bumps into this thing in this particular state, and these two things indicate its state, if this thing bumps into this thing, then they will form a complex. And I'll just write a lot of rules like that. Now, you can well imagine what kind of reasoning a system like that does. Basically, you simulate dumping large amounts of stuff into a mixture, and you shake the mixture, and you see what reactions happen. And that's the sort of level at which Walter works. It's very much more mechanistic. It is still not at the level of the shapes of the molecules. Right? So, for example, you couldn't design drugs this way. Because to design a drug, you actually have to understand the shape of the molecule so that you can get something to sort of get in there and bind to it. So these binding sites are actually abstractions, as we computer scientists would understand them. Right? There's a level of abstraction that's implicit in this kind of modeling. But it does give you something about the kinematics of the reactions, because you can affect the amount of stuff that's in the mixture. Right? You can increase the amount of one kind of molecule, decrease the amount of another, shake more or less vigorously, and be able to understand something about the dynamics, kinematics of the mechanisms. Here's another kind of model, different lab at Harvard, Peter Sorger's lab. Peter actually views biochemical processes as programs. And he imagines a software stack just like any other software stack in which we make good use of hierarchy, modularity, and abstraction. And so down at the very bottom here is the sort of thing that you just saw. It's very, very fine-grained molecular interactions. But then we sort of abstract up to the kinds of rules that I showed you about what binds to what, except that these ones are a little bit more complex, a little bit more abstract, and up to various kinds of macros and what they call motifs, and then modules that talk about entire systems, like an entire pathway, for example. And Peter and his colleagues have developed a programming language called PySB for writing that kind of code. So this one has to do with transportation across pores, and there's a little red box there that talks about how anti-apoptotics bind, and if you expand the function that's in that thing, you get to the definition of the function, and after you go through all of the documentation, you end up with something that actually tells you how that binding is going to work. It's in some idiosyncratic language that the biologists understand, but basically you're writing programs to simulate these biological processes, and that kind of many levels of abstraction give you all of the advantages that we know and love from computer science. And if you compare it with what people used to do, which is go from some conception down to ordinary differential equations, of which there may be thousands, and you change one and you have to change all of them, you're clearly better off than you were several years ago doing that kind of modeling. Peter calls this executable knowledge, and I'm going to come back to that a little bit later. I think this idea of executable knowledge is really important, because what we publish in journals today is not executable. Right? But if in fact you're encoding what you know about these systems as a simulation or as code of one kind or another, that's what you should be publishing. There are other kinds of models out there. I showed you a moment ago in Kappa and in PySB models that actually run in some sense. They generate stuff. There are other models that are essentially markup languages that don't generate stuff. One of them is BioPax. It's very, very popular. It's an XML-based language for biocuration. In BioPax, you can say MEC forms a complex with KSR1. That complex phosphorylates some sort of potentiates some reaction that involves these things. You can describe the reactions that are happening in this form of XML. It does not, however, generate those reactions itself. People have built simulators to sit on top of it. There are, however, no rate constants. And while in BioPax you can identify how many sites there are for binding, it doesn't identify particular residues, that is, places on the sequence that the binding actually happens. Even less information is carried by something called Bell. In Bell, basically, you can simply describe what happens. So this thing here says that this complex potentiates transcription. That's all you can say. One thing increases or decreases another thing. So all of these languages are sort of floating around, and they've all been developed by biologists for doing systems biology. And the question is, which do you want? What do they provide? What kind of understanding of the underlying mechanism do they provide? And that led us all to the following question. You know, we say we have a big mechanism program, and our job is to build mechanistic models, but what is a mechanistic model? Given that it could be in a variety of different forms, and it could have an interpreter or not have an interpreter, it can have syntax and a semantics. And the best we've been able to come up with, and I think it's sort of a little unsatisfying, but this is sort of where we're at right now, is that ordinarily when you think about modeling, you think about a function f of inputs that produces y as outputs, and we put a little hat above the y as a sort of a marker of our pragmatic intention that the y's that are predicted should match the y's that actually happen out there in the world. Right? So whenever you see a hat, right, it's an estimate. Right? And any model of the form f of x is what goes to y hat marks a pragmatic intention to compare the predictions of the model with the world. What's a mechanistic model? A mechanistic model is one where there's a hat over the f. In other words, we're making a claim that the function that we have proposed produces those y's is a good estimate of some function out there in the world. Pragmatically, we're making a claim about mechanism. Pragmatically, we're making a claim about how the machine works. And the reason I go into this is that people often ask me, well, isn't this a big data program? And I say, hell no. It's not a big data program at all, because big data is concerned with the deviations between estimated y's and actual y's. And this is concerned with the deviation between estimated f's and actual f's. Big data is all about correlation. Big data is all about association. It's not really about mechanism. And here we're trying to be about mechanism. So a mechanistic model is one in which you're making some claim about the mechanism. And a big mechanism we define to be one that is predominantly made up of those sorts of claims, but not exclusively. And the reasons not exclusively are very straightforward. We might not actually know how f works. And this is often the case. I mean, we reach the limits of our knowledge. We say, I can give you a transfer function that takes you from x's to y's, but I don't actually understand how it works. So some part of a big mechanism will be just those sorts of transfer functions. Another reason that we don't necessarily make claims about f is that we just don't care. We leave it to someone else. It's outside our area of expertise. Right? And that often happens when mechanisms are hierarchical. When I study things at one level of a hierarchy, for example, the person who's actually looking at the structure of the molecule, right? And somebody else studies the mechanism at a higher level, say the binding sites, without thinking too much about conformation. Right? So not all Fs in big mechanisms will have hats over them, but largely they will. We're trying to figure out causal knowledge of how the mechanisms work. Which means that languages that have been designed for markup of biological results, like Bell, aren't mechanistic at all. Whereas languages like Kappa, in which you can turn the crank and see the mechanism emerge, are mechanistic in the sense that they produce mechanism, right? But the language itself may not be mechanistic. And then languages like PySP, which is actually a programming language for building biological models, is mechanistic in exactly this sense. You're actually writing the mechanism. That all took a whole lot longer than it should have. I mean, we're 7 months into the program, and only now do we have anything like an answer to the question, what's a big mechanism? I'm a little surprised, but I guess people hadn't really asked the question before. And so, in a way, I'm sort of happy that we've gotten at least this far. All right, let's talk now about reading. So, here's a sentence. RAS acts as a molecular switch that's activated upon GTP loading and deactivated upon hydrolysis. Why is that sentence hard to understand? There are a bunch of language people here, and they all know, but for those of you who don't know why such a sentence is hard to understand, well, here are a few of the reasons. You want to map that sentence into some semantic representation. Well, which modeling language are you using to represent the underlying biology? So one question that arises immediately is, what's the target semantics for the sentence? And as we've seen, it could be a bunch of different target semantics. Then, straightforwardly, we have to figure out what are the entities in that sentence. They are RAS and GTP and GDP. And we have to make sure we know what those things actually refer to, which means we have to be able to go out to standard biomedical ontologies and find them. Well, sorry, but biologists are really, really sloppy about how they name things. So that in and of itself is a problem. Then we have to understand that some terms are actually not real things. They're sort of stand-ins for groups of things. RAS, it turns out, is a family of proteins. So when you read RAS, you're not entirely sure what they're talking about. Are they talking about the whole family or some subpart of the family? That's a problem because biologists, like other academics, make claims that go well beyond their evidence. So they'll say RAS when they mean KRAS, right? Because that way the paper looks beefier. If it sounds like a cynical comment, it isn't. It's a technical problem, right? The problem is to figure out what people are really referring to.
Out of curiosity, when somebody reads that paper, how many of the intended audience understand that they mean KRAS?
It's generally somewhere in the paper. It may be nowhere near that sentence. And so one of the projects that we have going on right now is to try and take sentences like that and see whether we can find in the paper where there's evidential support for such a claim. Yeah. Then there's, classically there's this problem of co-reference. So the second mention of GTP is actually the same thing as the first one. You wouldn't know that. I mean, any human knows that. Machines have trouble with that kind of thing. Words like that refers, in this case, to the molecular switch. So standard reference resolution has a little bit of a problem because there's chaining going on. That refers to molecular switch refers to RAS. So there's inference involved in solving the problem. We have to understand that there are events being described, activation, loading, and hydrolysis. But those are not specific protein-protein bindings. Those are sort of general classes of events. And then we have to put entities in their appropriate roles in these events. And then we have to weave the events into a story. And the story has to be in one of these representation languages. And then we have to relate the story to everything else we know. So there's quite a lot of work going on in reading in the program. And as you can imagine, as a risk mitigation strategy, we're looking at a variety of different kinds of approaches, and they go all the way from extremely low-level information extraction, the kind of thing that's done by Elsevier on their collection of corpus of journal articles, all the way up to actually very deep, semantically motivated reading. This is from James Allen's work at IHMC. James is able to get out of a sentence not only the semantic roles, but also tense and modality and aspect and nominalizations. There's an awful lot of nominalization that goes on in the biology literature. So a verb, to phosphorylate, becomes aphosphorylation, right? Which makes the language a little hard to understand. Anyway, James is making great strides on very deep semantic reading of these sentences, sort of in between. Those of you who know about the abstract meaning representation that's being tied quite closely now to VerbNet and FrameNet, we have a project going on for a biological version of the abstract meaning representation. It is less deep, I think, than the one James is doing. So we're sort of all along this spectrum from sort of grungy information extraction all the way up to deep semantics. I'm going to tell you about two kinds of evaluation that we've run in the last few weeks. I'm going to skip very quickly over the shallow evaluation. This is recall and precision of the various systems on being able to identify these entities in text. We're basically doing state-of-the-art when compared with other things that extract biological entities from text. You see up at the top biologists from Harvard who did this as a sort of a volunteer exercise. They turned out to be our gold standard, and a couple of the systems are sort of approaching their level. For event extraction, people are not doing quite as well. Events are harder to extract than entities. But again, as far as we can tell by looking at other competitions that have run on this kind of evaluation, we're doing pretty much state of the art. But here's what I really want to talk about. One of the innovations of the program is what we call reading with models. Now, the idea that you would process text with something in mind has been around for an awfully long time. It's just people don't do it. Since the start of the statistical revolution in AI and natural language understanding, everything is a pipeline that goes from words to whatever target representation you have. Nobody ever says, well, wait, why was I reading? What was I reading for? And how could that affect the pipeline? Which seems kind of broken to me. I mean, I'm old enough that I remember what words like top-down actually meant, right? So this all seems like it could be improved. And so by reading with a model, we mean the following. Why are we reading? We're reading because we want to update our knowledge about biological mechanisms. We have some knowledge about biological mechanisms. We should use it to help us read. And so the question that we ask in the deep reading evaluation is, in what relations does the text stand to what I already know? I don't want to complete everything out of the text. I want to know, what does the text tell me about what I already know? And so here are some examples. If you know that Ras binds RAF, then the text, monoubiquitination of Ras enhances association with downstream effectors of Ras, actually doesn't tell you anything you don't already know. So the relation between the text and the model is a corroborating relation. You have additional evidence for something you already know. Here's another example. Same sentence, but if the prior model doesn't talk about the downstream effectors, then you actually learn something new. It's a new mechanism, Ras binds P3 kinase. So again, the relation that's being extracted is a new mechanism relation. Some new mechanism has been identified. Not everything is mechanism. Some of it is relation. So enhances transcription is not mechanism. It's simply telling you there's some relationship between the amount of this and the amount of that. But again, if this is what you know, this text tells you that there's a relationship between Ras-ASPP complex and the transcriptional activity of P53, that is this protein that probably many of you know is associated with a lot of cancers. In fact, with all cancers as far as I know. So by deep reading, we mean tell me what the text tells me about what we already know. Yes?
So don't you also think that we're also doing the other way around? So we're asking, let my model tell me what it can about what the text is saying. So the corroboration is not just this corroborates something I have in my model, but it's my model corroborates this statement in the text, which is not necessarily, authoritative. I mean, like, we're not sure that the text is true or not. So when we see something that that agrees.
No, it's a very, very good point. And in fact, it's the sort of, that point is at the heart of my response to the following question. How do you establish the validity of the stuff that you're extracting from papers? And typically the way people answer that question, and certainly the way they answered them in all of the proposals we got to the program, is, well, if it's from Harvard, it must be right. Or Hopkins, sorry. We'll look at provenance. Your answer is a much better one. If big mechanism actually works, every assertion gets thrown up against an enormous amount of stuff. And it fights it out with that enormous amount of stuff. Right? If it's completely at variance with everything else we know, do you throw it away? Hell no. You say, that's really interesting. Right? If it corroborates everything you know, do you throw it away? Well, no, but you just say, well, it's not that interesting. So you're right. I mean, the fact that the model can help you establish the utility and the validity of what you're reading is very, very, very important. Yeah.
So to really do this, you want to look for evidentiary support, right?
I beg your pardon?
To really do this, you want to look for evidentiary support.
We do. And so we have a couple of projects going on now to find the parts of the text that provide evidentiary support. And there are two kinds. One is where we actually talk about experimental results, and the other is the weasel words, right? And we're looking at both of them. So what you will see is this experiment suggests that, which means I'm not really sure. And a knockout experiment is a very different kind of thing, the one that simply finds an association between increasing this and increasing that. So yes, very different kinds of evidential support.
There's also support by citing somebody else's work, and you don't want to double count if that work is already in your database.
No, because these days people make these sort of obligatory citations that have really very little to do. In fact, if you go and look at what people are citing, it often has no discernible relationship to what's being described. So you're right. I mean, there's a big chunk of this project that has to do with what I guess you would call the sort of sociology of academic publishing, which, wait. Anyway, deep reading results. Much harder task. We do less well at it than we do at entity and evidence extraction. We're actually not very good at identifying new relations. Red and orange are good, blue and green are bad. Not very good at identifying new relationships between text and what the model says. But that's because we don't have much practice, because machine reading has been dominated by data-driven methods. Once we actually get good at reading with models, I think you'll see these numbers change. Yeah?
So which of the representation are you using, the parallel or the PySB or parallel, to see which model is? So whenever you read, you have to then map to the model.
Yeah.
You said not all schemas would have already.
Yeah.
So which of the schemas did you?
That's a great question. So the people at MITRE who we hired to do this evaluation hate me because I required them to do it in all 4 languages, all 4 modeling languages. And they scored appropriate to that modeling language. So it was actually a very painstaking evaluation, and actually the sample size is small. We only, for this, we only looked at 6 paragraphs, 6 specific paragraphs, because the amount of effort that went into scoring was just prodigious. Okay, I'm going to very quickly skip over what happens after reading, which is this business of assembly. And the reason is that we just haven't gone very far in it. There are sort of two main approaches to assembly, and I will tell you I'm not happy with either of them, and I will tell you why. So one approach to assembly says, if up here we are plotting all of the interactions that we find in the literature between one thing and another, and we are trying to account for experimental results in a Bayesian manner, then we could probably reduce that thing up there to something simple. And so Richard Grower at SRA has developed an algorithm to do that. And that's nice. We call it reducing the hairball. If you see protein-protein interaction maps, they're just horrible hairballs. It's really nice to reduce them. What I don't like about it is that this is not a biologically inspired algorithm. Basically, it's using some minimum description length criterion. That's nice, but nature is well known to not respect Occam. Right? It's not clear that what comes out of this has much correspondence to the underlying biology. So I don't like it very much. The other approach, which you've got to admire its ingenuity, says, let's take the model fragments that we have and then some AMR representation of text and propose a new model in a standard sort of Bayesian way that says, look, the probability of a new graph given the AMR is proportional to this likelihood function, and we'll make some assumptions to make it tractable. What's not so good about that? It's going directly from text to model with nothing in the middle. There's no point at which somebody says, is that a good idea? Does it make sense biologically? It's saying, the text says this, so we will update the model. Yes, you had a question.
Yeah, so the picture that you have is just showing graphs, but really these are distributions over graphs?
In fact, relates to distributions over models that include RAS. So there can be thousands of these graphs.
That's fine, yeah. I was talking about the blue equation at the top.
Yes, yeah.
Where you're doing the Bayesian update.
So this is absolutely over distributions of models.
Yep.
Yeah. So basically all of these people are saying we have this essentially infinite pile of graphs. We prefer one graph to another. Right. There's some probability assigned to any one of to those graphs, to any one of those graphs. All right, let me wrap up. Big Mechanism was not supposed to be a cancer biology program or a machine reading program. And so one question that we're asking, even at this early stage, is if this idea of push scholarship and building big mechanisms for humans to understand complicated systems is going to work, we have to be thinking about what would be required to build such a system in another domain. And that's this brass ring that we're all stretching for. And I don't know how to do it, but there have been some sort of interesting things that have come out of the first 7 months. The first is we quickly gave up on the idea of the one true model. Now you can see how you could have this crazy idea that there's one true model, because everybody publishes one model, right? But then you step back and you realize, wait, if everybody is publishing one model, but they're all different, then we need to be looking at distributions of models, as you're saying. And so we need ways of organizing those distributions of models. Pretty much everyone has sort of settled on a kind of a rule-based language for describing these models, which you can think of as sort of nested boxes. If I were at this point to try and write an algebra for the kinds of models that we're seeing in Big Mechanism, the algebra would say a box either contains a transfer function that takes you empirically from inputs to outputs, or it contains other boxes connected in various ways, in various relations to each other. And that's irrespective of whether you're doing Bell, Biopax, Kappa, or PySV, this sort of nested boxes, rules that implement nested boxes seems to be the norm. We talked a bit about this idea of what's a mechanistic model. We've pretty much agreed that even if nature is not modular or hierarchical, humans need hierarchical modular architectures to understand the mess. So even though it's sort of a fiction, it's a fiction that we promote. We're pretty sure that reading can be improved dramatically over the current state of the art by reading for assembly, or reading, basically what I call earlier, reading with models. One interesting thing to have come out of the information extraction work is that there appear to be sort of roughly 10 ways to express any biological relationship. Is that something that's sort of inherent to biology language, or is it generally the case that for any given verb there are only sort of 10 different ways to express it? We don't really know, but that would be interesting. It would really speed things up. We are emphasizing precision over recall simply because we're awash in publications and we can afford a recall penalty. Yes?
When you say biological events, do you mean specifically protein-protein interactions? Or something?
That, but also processes just like tumorigenesis or apoptosis. I mean, yes, we're focusing largely on protein-protein interactions, but another thing that should probably be on this slide is that humans have this amazing ability to work at multiple scales simultaneously. So in one sentence, you'll be talking about this binding site on a molecule and apoptosis, which is what, 7 orders of magnitude? I don't know. I mean, so we're sort of, any given sentence can sort of bounce around these levels of abstraction like crazy. And so on. All right, so I think one of the reasons that the program has proved to be more challenging than we thought it would be is that what's actually happening is we're starting with text and we're going through multiple levels of description before we get to what we call mechanisms. And this stuff in the middle I call purgatory. And we haven't really figured out what's going on in purgatory. We know that as we move from here to here, we are also moving across levels of linguistic abstraction, biological abstraction, and mechanistic abstraction. And we know that some of the things that happen in the middle are, well, as you go from the text to, say, a model in Bell, it's a lossy transformation. So we know that there's some of that happening. We know that a lot of inference is happening, where biological knowledge is being brought to bear to take an impoverished text and produce a model, and so on. So one of the things I think is really interesting about the program that we haven't anticipated when we started is that you don't just go from text to biological model. You go through these intermediate descriptions. And I think we haven't really plumbed everything that's happening in those intermediate descriptions. We do call it purgatory, in which one undergoes purification so as to achieve the holiness necessary to enter the joy of heaven. But the interesting thing about it is, how do you know that it's ready to enter the kingdom of heaven? Right? I mean, by what standards are these re-descriptions of the text being judged? And I think we've only started to think about that. I started with a picture of where we're trying to go in the big mechanism program. I said that I don't think that there's ever going to be one true model. So the question I really want to leave you with, which seems to be a new question for science, is if you have a very large number of models, tens of thousands of models of complicated processes, how do you manage them? How do you publish them? Who owns them? What kind of reasoners operate over them? Think about a future in which there are tens of thousands of climate models, tens of thousands of models of cities or economies. I have absolutely no idea how to organize that stuff, access it, merge models. In a way, it's a bit like what goes on with Stack Overflow, except in Stack Overflow, you just have the pieces, right? Nobody's trying to maintain multiple systems built out of those pieces. So it's great that we have people now pushing mechanism out, as you see in Stack Overflow. But any given mechanism will have lots of versions, and they still have to be brought together into a model, into what we would call a system. So I think those problems, I think we're, as far as I know, we're just at the very threshold of understanding those kinds of problems. The last thing I want to say is of more personal nature. So I've been just knocked out by how outward-looking and interdisciplinary computer scientists are at Hopkins. That is not true in all computer science departments. Right? Academic ultra-specialization, introversion is still the norm. This is Google Ngram tracker. These curves are scaled to make them easier to see. But basically, this is the occurrence of the word Sputnik. And look what happens to liberal arts as a term in the vernacular. Whereas the term expertise, multidisciplinarity, polymathy, interdisciplinarity, to me what these curves are saying, you can interpret them any way you like, to me what these curves are saying is that this much vaunted goal of interdisciplinarity is identifying a problem, not a solution. Right? The word is in the vocabulary because we have a problem. And the problem is ultra-specialization. Now, as an academic, that really bothers me because I think we're doing science wrong. As a DARPA program manager, you can say, well, all right, let's develop technology like big mechanism to support another way of doing science. But it's actually the job of the universities to inculcate in students the understanding that we've got to understand big, complicated systems. And that sort of going into your office and studying one tiny little thing for your entire life, when we depend on power and energy and food and economies and God knows what else, and these are big systems, is just, we're not setting ourselves up for success. So with that, I thank you for your attention, and I'll take any questions. Yeah.
I had a ratcheting back from your last slide, going back to more of the big mechanisms. I was wondering whether the whole idea of building this mechanism up as you get more and more text or other forms of information, whether it doesn't have an intrinsic trap, which is that you're not going to revisit all the little pieces of information thrown at the mechanism every time one new one comes in. You'll presumably do something incrementally. And then wouldn't you get into a mechanistic model which gets more and more and more complex because way in the beginning you made a wrong move? Yeah. And the classic example is, the old Ptolemaic universe where you had to keep adding epicycles, and there were a couple of papers in my field about it. And then eventually along comes Kepler and says, nope, scratch all this, and then all of a sudden.
Yeah. No, it's absolutely right. So this is, of course, an old problem. It's the problem of non-monotonicity, right. And there aren't many good solutions to it. The good news, however, is that we can afford to store a lot of models. Right. We can afford to have a lot of variance.
So we wouldn't make Galileo recant. We'd let him be.
We'd let him be. Yeah. But of course, that's brave talk, right? I mean, yeah. What are you going to do with 10,000 models? I mean, nobody wants 10,000 models. You want to know what's approximately right. And so I don't really think we have an answer. I don't really think we have a solution to the problem. Clearly, these models will change. That's why we're reading. But it's a significant problem. Yes?
So I have a sort of very high-level question related to your last slide. there's a knowledge that each of us has inside our heads, which I think is fragmentary, contradictory, reflects multiple ways of framing the same stuff. Nonetheless, we managed to, we've got some knowledge representation scheme which supports reasoning under these fragmentary conditions. If we understood a way, if we had a way of describing what you have in your head, would that scale up to describe what the scientific community has as a whole, and to do reasoning and modeling at a large scale? In other words, is there a cognitive science answer to the question you have on the slide about how to organize academia?
Oh, I didn't realize that's where the question was going, but it's a very good question, isn't it? So since we know that humans can't read the entire damn literature and yet they muddle through, what are they doing that machines should be emulating?
That's right.
A great question.
And I don't think the question is limited to scientific understanding.
No.
I mean, if you, it's fascinating, reading comment threads on, say, the New York Times or whatever your favorite publication is, and seeing the different ways that people, frame issues and struggle through and so forth. And I believe that everybody understands all the framings.
Yeah.
And they're somehow drawing conclusions which are certainly fallible.
Well, one reason I think that Sorger's work at Harvard in this sort of software stack is interesting is that up at the higher levels, you have what are called motifs. And there's sort of ways that reactions can happen. And I suspect that that's probably the kind of thing that an expert biologist brings to reading a paper. And maybe it's that kind of thing that's more cognitive. I don't know. Certainly, it wouldn't be very human-like to be always at the very, very lowest level of description of any process. I think we make heavy use of abstraction. So whether we're doing this here adequately, I can't say.
So I guess the sort of issue as a scientist that I have with thinking about this in this way is that it seems to me that the breakthroughs happen basically when somebody who understands the literature realizes that one of the premises of all the experiments sort of left something really important out. And if that's true, then it's not going to be sort of present in the literature as you see it. And so I guess how does a system like this sort of reason about what might be missing that needs to be investigated further, which seems like maybe the more useful, or at least a useful outcome, like sort of to suggest sort of where the dark shadows are as opposed to trying to make a model out of what's there.
Actually, that was one of the use cases that people are most excited about in the program, but it probably works better at some levels than others. We know, for example, that there are so-called paradoxical results in cell signaling, where you push the button and you really should be seeing something light up over here and you don't. And so you know that the model is incomplete at the level of these molecular interactions, so the hunt is on for why. Or you suppress this and this just keeps on firing. Why? So at that level, identifying holes, I think, is a really good thing to do, and it's likely to be very, very productive. But then at a higher level, it gets back to what you're saying. Changes in fields often happen because people reconceptualize the field, right? And that we don't know how to do that. We don't know how to sort of say, I know how you used to think about this, but think about it this other way. Think about this as a control problem, not as a signaling problem. Okay, that changes everything. It changes the way people write all of these papers. It changes the nature of our models. We're nowhere near machines being able to do that. Yeah. Yes?
So, you brought up Stack Overflow example, and so one of the things that happens there is in the questions, people have seen a lot of times in, let's say, Math Overflow or biology or Stack Exchange, people will ask questions and then the answers are really just like somebody would know the relevant literature and just be able to pull up people would ask questions in very general terminology, and then other people would be able to find what specific instance of the paper or how it should be translated into the right terminology. So are you also building a map of the different ways in which people refer to?
Yeah, let me paraphrase the question and answer it. as follows. This business of mapping a question to some knowledge that's being acquired for some other purpose might well be an Achilles heel of this whole program. And I really haven't gone very far down this path, but it goes something like this. When I do an experiment, I have a reason for doing it. If I'm not capturing the reasons that I did the experiment, if I don't have that context in this kind of thing, all I have is that this influences that. Then it's going to be difficult to know how to answer a question that has to do with mapping a result to a different context. Right? And so I've been wondering about this because there is a sort of a very just the facts please aspect to this project that kind of bothers me. A good example of that is the reduction of a great big hairball to a much smaller thing. In effect, that means throwing out a lot of knowledge that people have acquired. And so you go to somebody who runs a lab, and they've derived this result. And you say, thanks, we don't need it. You know, you tell us that this influences that, but actually I don't need that in order to predict the data that I've observed. So, and the person who did that experiment says, well, that's all very well, but you have no idea why I did the experiment. You know, if you knew why I did the experiment, you would see that you can't just throw the results out. So I'm worried about that. I'm worried about the context in which the work was done and how we capture the context. I don't actually think the sort of saying, just focus on the results and forget everything else is the right way to go. Anyway, we're out of time. Thank you very much for your attention and for your excellent questions.
Introduce you here. Jason, Ben, and back there.
Great, thank you. Thanks so much.
Sometime after my Thank you.
Oh, thank you.
Hey, Jason. Very close. Jason, the reservation's in your name.
Oh, I thought it was impossible problem. That's what it said.
Oh, you called me. Okay. Well, I thought you are hosting.
Yeah, well, that's fine.
But you can like imagine like looking
So you're Jason.
I'm Jason. Yeah, nice to meet you. Thanks for talking about it. I'm surprised.
That's true.
You got 15 minutes.
So when you get to my roommate, wow, you're yeah.
Oh yeah. I was entertaining for a period of time. It lasted very briefly. But like they had some really creative descriptions.
No hard off.
Yeah, I can read it. Right, pencil.
The very professional days. Let me sharpen my pencils.
His uncle. Like, district master.
No.
floor are you going on?
2.
Okay.
How are you doing?
A number of issues. The microphone didn't seem to be working at the very end. I saw the audio here going green, and then there was no audio here. So we decided to go with the camera again. And then the speaker kept this microphone on, even though we're doing the camera recording, the audio source was a camera. He kept the microphone on him. And at the very end of the session, I look at this number and it changed. From A0, it went to A8. I'm not sure what was going on.
As far as were you having trouble hearing him, this is all looks like it's all the way up on the positives, so you shouldn't have a problem. We'll have to look
The thing
into
is, it was here in the bar. You couldn't hear. Now it's the camera. No, this is frozen now. The screen is frozen. I cannot stop it. I'm waiting for it.
Okay.
But yeah, the audio wasn't going through the panoptone. I'm not sure if it's not supposed to be there. But we disconnected this and connected it back, and we couldn't see the audio going out. So, but the weirdest thing was the channel changed.
without touching this at all.
No, I'm not. So the speaker had this in his pocket. I'm not sure if the buttons are being pressed when you have it in your pocket.
Well, now this is saying A0.
And this was a.
Maybe they were communicating to each other. Of course, this is off now.
Well, yeah, maybe
I mean, when
it's
you
power.
when it's off and then you turn it on, I don't know how that's going to affect it.
Well, we weren't recording from this, but I noticed the channel that changed.
Yeah, I don't know if it's going to be smart, and maybe both of them will switch. I'll have to look at the manual and see if that's the case. But if it's not picking it up here, then we want to rule out which one's the issue.
Yeah, it didn't.
I don't think it's. This should be a pretty solid connection.
Yeah. We can. I don't know if we can run a test. I'm kind of curious with this Panopto because it's frozen. I hope the recording is saved. Can try turning it off. I have a laptop here. We can try that. We can test the microphone.